Assalamu alaikum viewers. Today, working with DNA. The discussion topic is restriction enzyme analysis using gel electrophoresis. We will discuss about the process of cutting DNA using restriction enzyme, and here we'll discuss about the enzyme. What is enzyme? Enzyme are protein that catalyze specific chemical reaction under a specific condition. Recognize how enzyme are used to make recombinant DNA technology here. Identify the restriction sites in double-stranded DNA. This is because DNA technology are used in a range of industries. Your bodies and your foods are made of cells with DNA and protein. If we are here to know about the everything like living organism or everything that are made from living organism is the basic component is DNA and protein is must inside here. Reminds enzyme. What is enzyme? Here we will discuss about some characteristics of enzyme that are very familiar. Enzyme are biological catalysts found in all living cells. It is a basic component because enzyme are the thing that drive or catalyze or drive the chemical reaction inside all the cells. They are molecules, usually protein, that speed up chemical reaction. If we discuss about the nature of enzyme, is these are proteinous in nature. They mostly consist of protein. Enzyme have unique chemical structure, and that means they only act on specific substrate. It is the best one characteristics of enzyme because enzyme act on specific site with specific substrate. Enzyme never act on randomly on every substrate because it's the best one characteristics of enzyme is that it's specific in function. They must catalyze only specific substrate. For a particular enzyme to work optimally, it must be under specific condition like temperature, pH. Otherwise, its unique chemical structure will be disrupted. If these situation suitable conditions like temperature, pH is not present, then enzyme will never work because their structure, their characteristics of working is totally disrupt and destroy their chemical nature. When it is the most familiar that when the structure of protein is disrupted or changed, then it will never work normally. Enzymes are critical for a range of cellular processes, including digestion, DNA replication, protein synthesis. These are some applications of enzyme here. It's worked so properly, like digestion enzyme, DNA replication, when we replicate the DNA genetic material, like protein synthesis. Enzyme also act like in digestion, like cell glycolysis, Krebs cycle, a lot of chemical process that must be drive with enzyme. Here we'll discuss about the restriction enzyme. What is the purpose of restriction enzyme using in this process in this life? Researchers rely on restriction enzyme to assist with many processes in laboratories around the world, making recombinant DNA and appraising success. Mostly scientists, researchers use recombinant DNA technology to find out the drugs of different diseases within different experiments. Here we'll discuss about something like recombinant DNA technology is the best one here. Example for research, medicine, agriculture, because through recombinant DNA technology we find a lot of medicine we use in many agriculture process. Like here we'll discuss about some examples like we have many examples like golden rice is the best one here. Next application is our purpose is DNA profile analysis for disease diagnosis, paternity, family relationship test, and forensic test, etc. So we can use restriction enzyme in different life process, in different sociological process like diagnosis, disease diagnosis, or paternity, some issues between parents for their children. We can say in family relationship testing, how to find the parents of a young one. Forensic tests like criminal activities. We can also find these relationship between some suspected DNA. through using restriction enzyme, the best one here to find out the best solution to find out the criminal and between suspense. Restriction enzyme. These enzymes were discovered in bacteria in the very first restriction enzyme found in bacteria. Then they help the bacteria destroy viral DNA. It's the best one here is a natural process that bacteria destroy bacterial DNA, like we can say here a plasmid. Plasmid is a bacterial circular DNA that destroy the viral DNA. Bacterial give versus 2. The cut between specific bases letters of double-stranded DNA. This is a very best characteristic is that restriction enzyme act on specific side to cut the DNA into different parts. Here we will discuss about the double-stranded DNA molecule, how to cut in different pieces. The DNA is then multiplied multiple pieces. When DNA is cut through restriction enzyme, then it converts into different parts, different pieces. In detail, we'll discuss in the next sometime. The pieces are separately by gel electrophoresis for analysis. Ultimately, restriction enzyme act through a specific way, a specific method to find out the solution of problem. Specific restriction enzyme cut at specific DNA sequence. It must be known that restriction enzyme is not a single enzyme. It's a set of enzymes that have the same characteristic, but they are specific for their specific site, specific substrate. Here, for example, E. coli is an enzyme that cut at this following sequence, GAATTC. Here in we can see here the GAATTC. We have a piece of DNA, and in this portion of DNA, we find that sequence like GAATTC. So E. coli R1 is specific restriction enzyme that cut only GAATT sequence. On other sequence, it not active. E. coli was discovered in E. coli bacteria. EcoR1 is discovered in E. coli bacteria. The resulting piece of DNA are called restriction fragment. When a piece of DNA is cut into different pieces, then we can call restriction fragments. Here many restriction enzyme. Here's some examples of restriction enzyme, like Hindle was discovered in H. influenza, and Pastel was discovered in P. strati. EcoRV was discovered in E. coli like bacteria, and 3-protruding N. What is 3-protruding N? Protruding N are called sticky N. Why might this be useful? Why these sticky N 3' ends of DNA is so useful? Here we'll discuss about in next slide. Yeah, here restriction fragment sticky N. What is the function of restriction sticky N? Sticky N is a portion of DNA that help to bind a specific site with the same sequence of nucleotide to the other DNA. Like here we can see here that is that blue color show here sticky N, and red color show here sticky N. Because both sticky N come together and stuck with each other, bind with each other, because they have specific nucleotide sequence. AATT have opponent sequence like TTAA. Ligation start, and then here recombinant DNA is formation occurs. So we can elaborate here. We have a vector. What is vector? We must know about the vector. And what is insert? What is insert? These are two terms used in this lecture. Vector are the carrier. We can say it transport genetic material from one organism to other organism. In green color, insert. What is insert? Insert is a specific portion of DNA. Is a specific portion of DNA. We can say like a gene that have specific sequence for specific character that are transported from one organism to other. Then in next organism, here's we will transport insert. It will perform their function. When you cut two separate molecules of DNA with same restriction enzyme, the fragment will have matching sticky end. This is now recombinant DNA is created. Here we look here, two color like recombinant blue, say red and green. This is complete recombinant DNA because here some DNA is a vectorized like a red color and insert like a green color. Booklet 3 part. For example, we have a piece of DNA. We can see here where the portion of DNA that contains a specific sequence for EcoR1 restriction enzyme action site. If we here look, then we find that at that site we found AATT. This is a site where restriction enzyme only act, and the next site is here AATTC. So what will happen there? Here, a restriction enzyme come and cut this DNA into three portion, three segment, because here two places where the restriction enzyme can act. Number of fragment will be 3 after cutting. The size of each fragment will be 12 base pair, 9 base pair, and 4 base pair. We can find this numbering through counting of DNA fragment.